We have Cole Brubaker from Civil Engineering, and his presentation is 3D Printing the Future. Do you need this or no?
Oh, I can just talk this way.
Okay.
Ready? All right, so I'd like to start off by having everybody think about the last time they drove across a bridge. So infrastructure is so common in our daily lives, we often take it for granted. I mean, when we step out of our houses to drive to work or drive to the store, we just assume that the roads and bridges we use are always safe and well maintained for us to use. Unfortunately, though, this isn't always necessarily the case. So just this past month, the American Society of Civil Engineers released a report on our infrastructure, grading it at a D+. So, in fact, our nation's bridges, over 9% of them are rated as structurally deficient. That adds up to over 180 million trips taken on bridges every single day on these structurally deficient structures. So, as students, we all understand the significance of such failing grades. It means something needs to change. And that's where my research comes into play. So, I'm in the Department of Civil and Environmental Engineering, and every day I go into work, I think about ways that we can improve our infrastructure and ways we can make it safer, more efficient, and more reliable. And I'm doing this through the use of various sorts of nanoparticles and quantum dots. So the questions that I want to look at is how can we make a material sensing system that's capable of detecting changes in the material state? And the simplest analogy we can come up with is through the use of a mood ring. So we all remember mood rings growing up. I mean, how else were you supposed to know if we're happy or sad or even in love if we didn't have a ring on our finger to tell us so? And I hate to be the bearer of bad news, but these rings were just simple materials that changed color in response to your body's temperature, not your actual feelings. So I want to take this idea of a mood ring and implement it on the large scale using 3D printing technologies with the unique optical properties of quantum dots. So, what is a quantum dot? A quantum dot is a tiny little nanocrystal on the scale of 1.5 nanometers that exhibits a unique white light characteristic. So to put that in perspective, if you pulled a hair out of your head, look down the diameter, a quantum dot would be 50,000 times smaller than that. So very, very small. And we're able to embed these in various sorts of materials and use them for sorts of sensing applications. So once we embed them in a material, we can expose them to external loads, and by monitoring changes in their optical characteristics or their color essentially, similar to a mood ring, we can determine in real time how much load has been applied to our system. So this offers a number of distinct advantages over traditional sensing technologies where you rely on physical sensors for data collection, which you then have to post-process, where instead we can just look at a material with a camera and we can determine how much load has been applied to it. So, I realize that going forward, the problems with our nation's infrastructure and these bridges won't be fixed overnight. They might not even be fixed within the next 10 years, but by doing research like this and focusing on the development of materials that can help prevent material failure, we should be able to develop the future of materials and our infrastructure. So by going forward using this 3D printing approach, we should be able to embed the safety and security of our nation's infrastructure by 3D printing the future. Thank you.
Thank you. Next up, we have Edward Dawson from the German department with Keeping It Real by Copying Copies, Media and Authenticity in Austrian Rap Music. Yeah, come on up. Are you going to use the mic?
Good morning. My name is Ted Dawson, and today I'm going to tell you a little bit about my research on Austrian rap music. Specifically, I'm interested in understanding how Austrian rap songs are able to draw together diverse media sources from different times and different places and use those to say something meaningful about Austrian culture right now. Doing this involves a lot of thinking about authenticity. And you probably know authenticity is a big category in rap music in general. And that's where we get this expression, keeping it real, right? Everyone's heard this, keeping it real. This is sort of rap's idea of what is authentic. And for many people, this idea of keeping it real can seem like a paradox, because we all know that rap music is about copies, right? Rap songs are made with lots of samples of other media, of earlier music usually, stitched together, and the texts are actually often created in a similar way with lots and lots of quotations, and yet these claim to be real. How can all these copies be real, right? The answer is, keeping it real is not about your sources. It's about the outcome of what you're saying, right? So if the way I put together things other people have said, sounds other people have made, if that can speak to what I really feel and what I really think about my current moment, that's real. And that's the really profound thing that rap music does, is it is able to say, by taking a lot of copies of existing media, we're able to say something about our lives right now. Austrian rap music does this exact same thing, but in a way even more profoundly, because Austrian rap is literally copying copies, right? Austrian rappers have copied the idea of copying from American rappers. And then a lot of their sources are American rap songs, so copies already that they are then copying. And when they do this, they speak to Austria right now. So this is an example behind me about an Austrian rap song from Linz from 1997. Linz is sort of the point where all the lines come together there. And what you're seeing is the way that this song pulls together lots of different media from different times and different places to talk about Linz in 1997. And I'm looking at a lot of other songs and looking at how they do similar things. And the reason I'm very interested in this, and that I'm interested in understanding how Austrian rappers are able to do this in all of these different songs, is because this reflects on the situation we are all in right now, right? We live in a society surrounded by media, right? Immersed, flooded by different media where we don't know, are they real? What's the authenticity of them? Where did they come from? We don't know the sources. And yet, using only these things, we somehow have to respond to the issues of our moment and reflect on them. And Austrian rap music can give us some guidance in how to do that. Thank you.
Thank you for that. Next up, we have Sanjay Mishra from Chemical and Physical Biology with An Eye for an Eye and Heart.
Thank you. So we all can agree, eyes are our window to the universe. But then all windows need glass. Where is the glass in there? You see that little white thing? That's the lens. And lens is the through which our universe is focused down to the retina, and we see what we see. Of course, what we see depends on what we want to see. But that's not the part that I'm interested in. I'm interested in how does that glass remains clean. Now, all of you who own a house, you know when your glass gets dirty, your window gets dirty, you call somebody and he cleans it for you. Who cleans this glass? And if it doesn't get clean as we age, as we get older, it gets cataract. We can't see through it. Now, being the protein, now this glass is all a lot of protein. Think of it as a very thick cheese, except it's transparent. There are no holes in it. Now, as it happens with all protein, think about your milk. You left it in the bottom of the freezer, bottom of the fridge. After a while, it curdles. That happens to most proteins, but these proteins have a weird situation. We are born with part of the lens which was put down within an hour of our birth, or our conception actually. And now, because being this thing, a transparent proteinaceous object, it does not have the ability to take those dirty proteins, sicken protein, and get them out. So how do we manage it? If you see this plot, all the lens proteins are generically called crystallins, and within those crystallins are the alpha crystallins and the beta gamma crystallins. The alpha crystallins are supposed to be the sponge who are looking for the sickening proteins, the beta gamma crystallins, and also their own kind, the alpha crystallins. And when they encounter these sick, dirty proteins, they encapsulate them around to make sure they don't form cataract. They don't form aggregates. And if you think about this, those aggregation phenomenon are at the center of not only cataract, but the Alzheimer, Parkinson's, CMT. And in fact, people believe that the reason we begin to age is because those accumulating sick proteins, they begin to kill other cells. So obviously, this process is central to a lot of what we understand about the aging and how proteins take care of themselves. This is where my project comes in. But think about this, we do understand that that can work, but how do we show it works? The easiest thing is, as the science develops, we begin to make tools where we can take one or the other proteins out of the system and see if actually things begin to happen the way we anticipated. So I work with zebrafish using a tool called CRISPR-Cas9. And we knock out these proteins, we make the zebrafish where the alpha A or the alpha B is now defective. And if it is defective, as you see in this plot that I've shown, as we had expected, once you take these proteins out, the alpha B1 in this case, or the alpha B2, which is another kind of alpha B, you begin to see that the fish which had those proteins missing compared to the control, they begin to develop cataract. Well, as it turns out, not only did they have cataract and the cloudy eyes, oh, thank you.
Next up, we have Amy Creecy from Biomedical Engineering with the effects of diabetes on bone matrix.
Hi everyone. Now, not everyone knows this, but when you have diabetes, you are more likely to break a bone. Unlike with a disease like osteoporosis, this is not because you have less bone. If anything, you have more of it. So why then does having a lot of sugar in your bloodstream, as what happens with diabetes, make your bones weaker? This is the question that my research seeks to answer. So since it's not a problem of bone quantity, then it's likely a problem with bone quality. To look at bone quality, I look at bone on the molecular level. Now, at this level, bone can be viewed as being made up of two things, protein and mineral. The mineral is what makes bone strong. It's like a ceramic. The proteins are what makes bone tough. The proteins are nicely linked together in this organization called the bone matrix. The links keep the shape of the bone, but the proteins can slide a little. Now, this may seem counterintuitive, but having this movement makes bone tough. It makes it to where it doesn't automatically break when it bends. It gives it some spring. Now, the idea is with diabetes, maybe all this sugar is messing with the structure of the bone matrix. When you have enough of it, sugar, or glucose, can attach itself to protein. It adds more of these links, and the bone loses the wiggle room that lets it slide. It just breaks. So looking at the bones of diabetic rodents, I've been able to see that we do get an increase in the sugar links and that the bones are weaker in some cases. Unfortunately, science doesn't always immediately give you straightforward, clear answers. Now, even with the bones that did not have an increase in the sugar links, the bones were still weaker, meaning I have a little bit more digging to do. Maybe it's not just the sugar links. Maybe the sugar is adding kinks to the protein. Maybe something's wrong with the composition of the mineral. Overall, though, the ultimate goal of this research is to figure out why these bones are weaker so that we can come up with a treatment to make them stronger. This may not seem like breaking a bone may not seem like a big deal when you're in your 20s and you're healthy, but it is a big deal when you're in your 70s and you can't bounce back right away, especially given all the other complications of diabetes. If we can find a treatment to make bones stronger, we can treat people so that they don't break a bone in the first place. This would improve people's quality of life, and which to me is ultimately one of the goals of science. Thank you.
Is it on? Okay, great. Okay, thank you so much. So I'm Laura, and I'm in the clinical psychology department at Vanderbilt. And today I'm going to talk to you about some work I've been doing in schizophrenia. So schizophrenia is an extremely debilitating mental disease that affects 1 in 100 people. It's been ranked as the highest healthcare burden that we have today. And just recently, the Surgeon General released a notice that social disconnection is one of the greatest problems that we're facing right now. So what we know about schizophrenia is that it is characterized by sort of a set of hallmark experiences and symptoms. Some of these are positive symptoms, so you can think of this as kind of things that happen above the baseline of a typical person's experience, so not necessarily good, positive, but just above the baseline. These are things like hallucinations, so seeing things that maybe aren't really there in reality, or hearing things such as voices that aren't there, as well as delusions. In addition, patients with schizophrenia exhibit negative or under the baseline of a typical person's experience. Symptoms such as social withdrawal, amotivation, and kind of just a lack of proclivity towards activities. But we also know that some of the major risk factors for developing schizophrenia are social exclusion, isolation, defeat, and social adversity. And so we wondered whether there was a potential link between all of these sort of exclusatory experiences that they might have faced and the later development of positive symptoms, which are highly social and often very emotional and distressing in nature. So what we find over and over again in our studies is not only are patients endorsing the fact that they're lonely and that they do desire social interaction, which has kind of long been this assumption that, well, they're anhedonic, they don't really care about being around people, they're just withdrawn by choice. But in fact, we replicate over and over again that this is not the case. Our patients rank their loneliness as very high and, in fact, much higher than controls consistently. And I've also been seeing in my work that not only are they endorsing loneliness, but this is related to how they do on social perceptual tasks. So people with greater loneliness are showing worse performance on social perception. So could it be that all of these things are linked together? What we wanted to do was try to find a way to sort of lessen the symptoms of loneliness and see whether that improved any other symptom outcomes. And so we decided to do this with a choir. Why choir? So singing together as a group has been shown in other populations to have this sort of special icebreaker bonding effect that allows people to bond quicker than other activities such as arts and crafts or other kind of groups together. We also know that singing activates the same parts of the brain that are hijacked during auditory hallucinations. And so we wanted to remedy that. What we found over just an 8-week choir was that they decreased loneliness, increased positive mood, and we even saw some improvement in cognitive deficits. Thank you.
Thank you. Next up, we have Daniel Kasima from Neuroscience with Drugs Are Sick, TLR4, and Reward Learning.
You want a snack, but you want to eat healthy. So you grab that bag of spinach from the vegetable bin. But a few hours later, you feel really sick. In today's polarized society, the idea that food poisoning is unpleasant may be one of the few things that we can all agree on. Nothing like a little E. coli to help tie humanity together. But often lost in this ordeal is another shared trait. We learn. We shape our behavior with experience. We might think, that spinach made me really sick. Maybe I should throw it out. Learning takes place in our brain. Our brain cells, called neurons, communicate with one another through structures called synapses. They form the language of learning. In place of letters, we have changes in the strength of synapses between particular cells. Like the alphabet is used to form words and sentences, many of these changes underlie learning and memory. In general, this is a good thing. If we couldn't change the strength of synapses, we wouldn't learn. Probably more of that spinach that made us really sick. But these same mechanisms of learning can also be used against us. You've probably heard that drugs are bad. Not everyone has a substance use disorder, but unfortunately, this condition also affects family and friends, meaning everyone here. Drugs like cocaine are the pirates of our neuronal language. They hijack our mechanisms of learning. They rewire our synapses to cause the foundation of addiction. My research looks into the interplay of learning, memory, and reward. But there's a twist. Does the immune system play a role? Is there a link between that E. coli in your spinach and cocaine? Turns out there might be. I used mice to study a protein called TLR4. It's one of our body's security cameras detecting bacteria like E. coli to sound the alarm. Normally, we change the strength of synapses with experience. It's how we learn. I found that mice that lack TLR4 have fewer ways to change synaptic strength. It follows that these mice may have fewer ways to be hijacked by drugs of abuse. In support of this, I found that while normal mice really like cocaine, mice that lack TLR4 don't. This suggests that TLR4 plays a role in learning, memory, and reward. And importantly, this opens up new directions and avenues to help those with synapses gone awry. So whether it's E. coli or cocaine, think about TLR4 the next time you think about drugs or food poisoning. Thank you.
Thank you. Next, we have Mikin Patel from Biological Sciences with exosomes, messengers in the brain.
The human brain still puzzles scientists. This is the headline from a New York Times article published in 1927. And after almost 90 years, this still remains true. And that is because human brain is a remarkably complex organ that contains over 100 billion specialized cells called neurons. To put that number in the context, there are almost as many neurons in your brain as the number of human beings ever lived. A typical neuron looks like this in the first image, with processes that resemble branches of a tree. Each neuron makes over 10,000 connections with other neurons through the structures that are mushroom-shaped called dendritic spines. And these structures are seen in the green staining in the second image. Communication between neurons at these dendritic spines is critical for higher brain functions such as learning and memory. Nearly 1 in 6 of world's population suffers from neurological disorders such as Alzheimer's, schizophrenia, autism, many of which are associated with abnormalities in the size and number of these dendritic spines. So how can we better understand the process of normal spine formation? In recent years, we have identified these tiny fluid-filled bubbles called exosomes that are being released from different cell types. These exosomes, they are like messengers or mailmen in our body. They carry information around and they deliver it to the right place and right time. We know that neurons release these exosomes, but we don't know why. And so my project is focused on identifying the role of these exosomes in neuronal development, especially looking at the formation of spines. And to test this, I targeted proteins HRS and Rep27B that are known to regulate formation and release of these exosomes. And then I measured changes in the number of spines. I found that there were more spine precursors called filopodia, as well as spines when I increased exosome release, while there were less filopodia and spines when I decreased exosome formation and release, suggesting that these exosomes, they carry message or information for the formation of these critical structures that are important for memory formation in our brain. So what is the message that they carry? How do they deliver this information at right place and right time? Can we use them in therapeutics by inserting missing information in the exosomes? As we have just started to understand the properties and functions of exosomes, there will be still many more challenges before we can use them in therapeutics. But I am hopeful. As Einstein said, learn from yesterday, live for today, hope for tomorrow. The important thing is to not stop questioning. Thank you.
Thank you. Next up, we have Douglas Clark from Religion with Radical Insider, Gayrod Wilmore, and the Black Freedom Struggle.
Was the civil rights movement the product of tactful, pragmatic reformers working within institutions and government and churches to implement gradual changes? Or was it the brainchild of strident radical critics of those institutions? Did it stem more from religious ideas about nonviolence and equality, or from Marxist and black nationalist calls for revolution? I argue that pragmatism and radicalism, both the religious and the revolutionary, were central to this movement, and I do so based on my research on Gayrod Wilmore, a scholar, minister, and activist. In the 60s, Wilmore was the primary official charged with promoting racial justice and civil rights for the United Presbyterian Church, a national majority-white denomination. There, he was an insider working closely with white moderates, church officials, and government leaders to promote demonstrations and pass legislation. Yet Wilmore was also a fierce radical critic of those same institutions. In that same insider role, just a few years later, still working as an official in a mostly white church, he championed black power and joined other radicals to demand that white churches, including his own employer, pay reparations for slavery. He even argued that black power and black radicalism actually were rooted in religion. Wilmore acted as sort of a mediator between moderate and radical wings of the movement. Why is this work important? The Moral Mondays movement of the North Carolina NAACP uses language of religion to build a large coalition of progressives, moderate and radical, religious and not religious. Some opponents of the Dakota Access Pipeline use religious language to demand that the government respect their sacred land and water, while others oppose the pipeline for environmental reasons. Also, some who mistakenly believe that the civil rights movement used only religious language have criticized Black Lives Matter for not relying more on religious rhetoric and institutions. My research on Wilmore demonstrates that the civil rights movement required the work of all kinds of people with all kinds of views about religion from a variety of political perspectives, those on the inside of institutions and those on the outside, as well as those skilled at mediating and building bridges between these different groups. History proves that then, as now, social activism requires all hands on deck. Thank you.
Thank you. Next up, we have Kate Snyder from Biological Sciences with Learning a Better Pickup Line, Do Birds Need More Complex Songs to Attract More Mates?
Two male songbirds walk into a heteronormative bar. The first one sings a light and delicate tune that roughly translates as, "Girl, I rise so early, I'll bring you only the finest worms for breakfast." The second just squawks a few times, which may roughly translate to, "Hey, girl." In this scenario, the first bird might attract a mate so quickly that he just keeps singing to try to attract another. The second bird just can't compete, and if he manages to find a mate, he might just have to keep her for life. So, As you walked into campus today, you likely heard at least a dozen different songs. What you're hearing are male birds trying to attract mates by singing the best song, the bird version of a cheesy pickup line. So, what drives these complex songs? That is a central question in songbird evolution. So, in our bar scenario, we illustrate that the complex song, like the one seen with the white-crowned sparrow, as you can see, that song has 5 different syllables, compared to the black-crested titmouse, which has 1 syllable, just repeated 5 times. So, in this scenario, I wanted to ask, with a different approach, to why do these things tend to cluster together? Since this hypothesis has been tested a few times, I'm taking a new approach. With the advent of DNA sequencing that is faster and cheaper, we can now build evolutionary trees like this one. I researched the traits of song and mating system of over 250 species by looking through a century of literature and overlaid the traits onto this evolutionary tree. As you can see, with the black and white representing mating system and the colors representing song syllable repertoire, the traits tend to cluster within the upper and the lower family groups. What my results show is that song does not drive mating system, or vice versa. In fact, instead, they are just co-evolved together just by chance when you can account for birds' ancestral history. It turns out that if you are polygynous and sing a complex song, you might just do that because your parents and grandparents did too. It turns out that in the tree of evolutionary history, birds of a feather really do flock together.
Thank you. Next up, we have Meghan Bowler from Biomedical Engineering with targeting cadherin 11 to prevent heart valve disease.
If you know someone over 65, raise your hand. That's pretty much everyone. Unfortunately, 1 in 4 of those people are affected by calcific aortic valve disease. This can eventually require total heart valve replacement because there is no cure and there is not another treatment. You may wonder, why don't we already have alternatives to total valve replacement surgery? And this is because we don't really understand the disease process. We do, however, know that an important protein in the development of calcification is cadherin 11. This protein extends from one cell to interact with cadherins on neighboring cells and relay important information to the insides of the cells. This works kind of like a handshake. The cadherin 11 hand grasps the hands of other cells, and the strength of the hand and length of the shake transmits important information between those cells. My thesis focuses on understanding how cells interpret that information of the handshake, or how cadherin 11 transduces mechanical signals into cell behavior. Now, as you age, your aortic heart valve stiffens naturally, and this causes the cells to contract more. This increases the tension between them, changing that handshake into more of a vice grip. Something unique to cadherin 11 interactions is that they're actually stronger than the integrity of the cell membrane itself. This means that cells can tear their neighbors apart limb from limb before letting go, which is not the kind of handshake I'd like to experience. This eventually leads to the development of calcific nodules, or all the bumpy regions you can see pointed out by white arrows on the disease valve to the right. Perhaps if we could disrupt some of these vice grips, we could prevent the formation of calcific nodules altogether. We tested this hypothesis in a mouse model of calcific aortic valve disease and saw dramatically improved heart valve function when they were given a blocking antibody against cadherin 11. This blocking antibody works by binding the outermost portion of the cadherin 11 protein, thus preventing interactions with other cadherins. In other words, it's as though someone's placed a baseball into your hand, preventing a robust handshake. As an added bonus, this particular blocking antibody is already in clinical trials to treat rheumatoid arthritis. This means a reduced timeline from our research to treating patients with heart valve disease. While there are still many mechanistic details of the how and why to clarify, blocking cadherin 11 interactions provides immense hope for establishing a non-surgical treatment or even cure for calcific aortic valve disease. And if you'd like to shake hands after the session, I promise I won't tear your arm off. Thank you.
Thank you. Next up, we have Elizabeth Lanphier, I'm so sorry, from philosophy with A House is Not a Home, but Why You Ought to Offer Hospitality in Each.
So try to imagine with me for a minute that it's 5 to 10 years from now. You live in a coastal city where you have a job you love, you've earned tenure, and you own your own house. However, sea levels are rising, and in the foreseeable future, your life might literally be underwater. Supposedly permanent job gone, house, property worthless. You and your community are forced to be displaced. What rights do you think you have in this situation? And what would you think others owe to you? Now, this might sound like a hard-to-imagine scenario, but due to climate change, individuals and communities are facing these very problems of displacement. One example is the people of the Republic of Kiribati. It's a constellation of islands in the South Pacific, none of which are more than 6 feet above sea level. And sea levels are predicted to rise up to 5 to 6 feet within the next century. So do the people of Kiribati have a right to become citizens elsewhere? And are we in the US, for example, obligated to welcome them in? If so, how might this infringe on our own rights to a sovereign nation state and territory? So you can see there's a tension between individually or collectively held rights to possessions, houses, land, citizenship, and our responsibilities to others, particularly the duty to offer hospitality and the duty to relieve suffering. In my dissertation, I argue that rights are not things we should own, even though there's a long tradition in the history of philosophy to understand rights as possessions. I'm going to call this the house model of rights. However, houses are things that we can own because we've established institutions such as those of private property. And often, these institutions are founded on and reproduce systems of inequality and injustice. So as an alternative, I want to offer a home model of rights. According to this home model, rights, like homes, are things that we form together, share in, and belong to, rather than things that belong to us. So according to this model, how do we resolve the tension between rights and responsibilities? I would say that the home model requires that we share even those things that we own, and that we understand rights as part of, not counter to, our sets of obligations toward others. Look, we can still own stuff, but our ownership is not our ethics. Instead, we should understand that justice is the act of participating and belonging together. So where does this leave us then with the climate refugee, whether it's the Kiribati people or our imagined or potential real future selves? I would argue that according to the home model, we have an obligation to share our common home of the planet. The planet and its territories are not houses that can be owned and controlled by those whose power, status, or luck happens to grant them possession. Instead, it's a home that we ought to share in together, be welcomed into, and to which we belong. Thank you.
Thank you. Next up, we have Krystian Kozek from Pharmacology with creating tools to study the link between G protein-gated inwardly rectifying potassium channels in the brain and drug addiction.
Good morning. Drug addiction and drug abuse are a big problem in our society. Millions of Americans suffer with people who have these diseases. Two common sources of drug abuse and drug addiction in the States are ethanol, also known as alcohol, and opiates. These two drugs kill over 100,000 people each year. 80,000 people die in alcohol-related deaths, and over 15,000 people die in opioid-related deaths. Opioids is what I'm going to focus on, and they're pretty ridiculous. Currently, the opioid epidemic in the United States has more than quadrupled over the last 10 years. It used to be in the 90s that 4 out of every 5 recovering heroin addicts first got their heroin or opiate addiction from heroin. Now, if you go to a clinic, 3 out of 5 people that are recovering from opiate overdoses or opiate addictions have gotten their first prescriptions from their physician. And so doctors are now the cause of opiate addictions. So when you break something, go get your medication, you are now at a chance, a significant chance of developing an opiate addiction. I think that's ridiculous, and I'd like to help solve that problem. And to do so, our lab studies a portion of the brain called the ventral tegmental area, and this ventral tegmental area is one of the cornerstones underlying opioid and general addiction processing. In this VTA, which is the ventral tegmental area, there are dopamine neurons, and inside these dopamine neurons, there's a very specific set of proteins expressed, and I study these proteins. These are the G protein-coupled inwardly rectifying potassium channels, and the subunits 2 and 3 come together to form a channel called the GERK2-3 channel. People have studied this in mice and found that if you get rid of the GERK3 subunit, you can actually cause mice to overtake and over-increase their abuse of substances. And if you if you overexpress this channel, then you can actually get mice that don't abuse substances such as ethanol, methamphetamines, and cocaine. With this in mind, we'd like to translate this research to humans. However, there's a problem. You can't actually remove or overexpress proteins in living humans that are, us. But what we can do is give people medications to achieve this same goal. So what I want to do is figure out if there are drug-like molecules we can give people or mice to study and change the activity of GERK2-3 and then change the activity of this circuitry that causes addictions in humans. So personally, I've gone ahead and screened over 100,000 molecules and found 9 molecules that can effectively regulate GERK2-3. We're currently working on making these into drug-like molecules that are efficacious in animal models. And to sum it all up, what I aim to do is to create molecules that will allow us to decrease the activity of the VTA and thereby decreasing the ability of people to become addicted and develop drug abuse. Thank you.
Thank you. Next up, we have MariaSanta Mangione from Cell and Developmental Biology with Ensuring Proper Cell Division, Insights from Yeast.
On average, our bodies comprise 37 trillion cells, though we started from a single fertilized egg. Through cell division, the cell number was able to increase exponentially. Every day, approximately 2 trillion cells divide for normal tissue maintenance, such as restoring dead skin cells, making new blood cells, or repairing wounds. And unfortunately, sometimes cell division goes awry, and this can give rise to diseases such as cancer or anemia. As you might expect for such a vital process, cell division occurs through a series of steps. The last step in this process is cytokinesis, which is the physical separation of the two daughter cells. In yeast and animals, cytokinesis proceeds through the assembly of a division apparatus, which is called the contractile ring. This ring constricts, pinching a single cell into two daughter cells through a purse string-like mechanism. Importantly, cytokinesis must not occur until after the genome is separated to the ends of the daughter cells. It's imperative for daughter cell survival that the genetic information not get damaged, so the placement of the plane of division is also very important. Two major questions in cytokinesis are, one, how is the timing of cytokinesis controlled? And two, how is the site of division selected? My research addresses these questions by studying a component of the division apparatus called CDC15. Prior to cytokinesis, CDC15 is inhibited through a superficial modification. This modification must be removed in order for CDC15 to participate in division apparatus assembly. My thesis work aims to identify regulators of CDC15. So far, I've identified 4 regulators. Loss of these regulators results in cytokinesis defects, although the cells are able to survive. These results are significant because the regulators might be controlling additional components of cytokinesis of the division apparatus. But also, it highlights the complexity of cell division regulation. It's such an essential process that there's multiple fail-safe mechanisms built in. My work will help us understand how a single fertilized egg can go and become the 37 trillion cells of a human adult. This in turn will help us understand human development, normal tissue function, and also the basis of various diseases. Thank you.
Thank you. And now May Shen from Psychology with The Quest for Visual Expertise.
Given an X-ray image, Mrs. Red, an expert radiologist, can diagnose Alzheimer's from a single look. In contrast, Mr. Green, an aspiring medical student, still has to guess. This ability that Mrs. Red has, the exceptional ability to make judgments with images, is called visual expertise. Beyond radiology, visual experts like Mrs. Red play important roles in many domains in our society, such as airport baggage screening and forensic fingerprint identification. My research goal is to understand such visual expertise so that researchers can develop more effective training systems for students like Mr. Green to develop their visual expertise. Specifically, I study what experts and novices see and how that relates to their performance. I use bird identification ability as an example to study these questions because bird identification is a commonly used and accessible example of visual expertise, unlike forensics and radiology. First, I study what bird experts and novices see in their mental space. In our mind's eye, we see things as points located in a multidimensional space. For example, everyone has a unique mental space for puppies, with dimensions such as the puppy's size, cuteness, and intelligence. In your puppy space, you might see your own puppy as ranking lower than your neighbors on the dimension of size, but much higher on the dimension of cuteness. And your neighbor would certainly have a different puppy space. Your personal puppy space would influence the similarities you observe among different puppies, providing a good metric for me to identify the dimensions of your puppy space. Using this logic, I measure and model bird experts and novices' bird space by gathering their similarity ratings among different birds. This will answer the question of what they see. Then I relate what they see to how they perform, in this case, bird identification performance. This relationship between what they see and how they perform has significant implications. For example, if there are dimensions in the expert's mental space that are critical for efficient diagnosis using X-ray images, which novices like Mr. Green are not aware of, we can highlight those dimensions in their training to improve the training efficiency. My name is May. My research is to help people with the quest for visual expertise. Thank you for your attention.
Thank you. Everybody, please join me in thanking our participants and our judges at this moment. So this concludes our first preliminary session, and we will now take a short break and begin the second preliminary session at 10:40.